Hey guys, Michelle Glass here, and welcome to our third episode in our Chapter 16, Lecture 16 series. And our topic for this presentation is the sympathetic nervous system. Remember, this is one of our two main divisions of the autonomic nervous system. So we are going to be controlling here our smooth muscle, cardiac muscle, adipose tissue, and our glands, those four possible effectors. Now, a lot of folks like to sort of use this catchphrase of fight or flight to help them to remember the role of the sympathetic nervous system in the body, and that is very much true. So if you are in a dangerous situation, that this is the nervous system division that will really start dominating and helping to make sure, hopefully, that you do survive. But it is important to keep in mind that this is not only active in stressful survival type situations, but this is a regular way of controlling your viscera. So we'll see it being active all the time, really. Okay, now when we are talking about the sympathetic nervous system, you do want to keep up with these four features of the sympathetic nervous system. So you should be able to, by the end of this video, describe how the sympathetic nervous system is organized. You should be able to talk about the functions of the sympathetic nervous system. So when we say it increases metabolism or it's responsible for fight or flight, you should be able to describe in much more detailed way exactly how it is controlling your body. What are the different effects of the sympathetic division? We will see there are special neurotransmitters released by the neurons of the sympathetic division. Neurotransmitters, remember, are chemical messengers that are released at a synapse. So it's a very localized, specialized sort of communication system. Okay, and then those neurotransmitters have to bind to receptors on that postsynaptic cell. And so we will see that there are special receptors that are involved in the sympathetic division. And because the neurotransmitters are binding to those receptors, there is a very close relationship there with those. Okay, as we begin with our sympathetic division, let's look first at organization. And so I have some key descriptions listed out for you here, and then we'll look at each of these in a little bit more detail as we go through. So if you remember from our second video, which highlighted the organization of the autonomic nervous system in general, we have two different types of fibers that we're keeping up with. We have the preganglionic fiber, which, remember, has its cell body located in either the brainstem or the spinal cord, and then we have the ganglionic fibers, which has its cell body located in the ganglia. Now, notice in the sympathetic division that our preganglionic fibers are typically very short. So they're located really close to the spinal cord. Their cell bodies are located in those lateral gray horns that you studied in A&P1, in the region of the spinal cord between T1, that's the thoracic vertebrae 1, and L2. L, of course, is standing for lumbar. So we will see the sympathetic division's preganglionic fibers housed, or the neurons are housed between T1 and L2. Okay, now we do see those preganglionic fibers are short. They're extending out to those ganglia. There are three different sympathetic ganglia. We have what's called the sympathetic chain, the collateral, and then also adrenal medulla, which is the singular version. Medullae with an AE ending would be plural, and we do have two of those, so you might want to pluralize it in your notes. And then the last feature of the sympathetic organization is that those ganglionic fibers, those fibers whose cell body is located in the ganglia, are very long. Okay, so the feature here is those ganglia, those three ganglia, for the most part, located right up there close to the spinal cord, and so that's what's allowing the preganglionic fiber to be short, but that's what's causing that ganglionic fiber to be very long. Now, whenever we talk about fiber, we're really just talking about the axon of that neuron, if you're getting confused with that. All right, we're going to take a look first at the sympathetic chain, and I have a beautiful picture for you. Ta-da! Okay. All right, so what we're looking at here is I've tried to kind of list out for you your brain stem and your spinal cord, and I've used the gold to sort of represent our ganglia. The ganglia, remember, are those processing centers in the peripheral nervous system. It's where the preganglionic fiber synapses onto the ganglionic fiber. Notice that our preganglionic fibers are in red, and our postganglionic fibers are in black. And so our preganglionic fibers have their cell body located between T1 and L2. And we'll see this will be different when we look at the parasympathetic division. Now you're looking here at what's called the sympathetic chain. And I like to think about this as like a beaded necklace. So you should think about those ganglia as extending actually all the way on either side of the spinal cord. And so it's called paravertebral, meaning para beside the spinal cord. So this is on either side of the spinal cord. And even though the cell bodies for the preganglionic neuron is between T1 and L2, those sympathetic chain ganglia go up above and down below that region. So it's a super long kind of beaded necklace. But it's close to the spinal cord, and so that means that those preganglionic neuron fibers are short. And then the ganglionic fibers are long because then they're going to extend to the different parts of the body. The sympathetic chain is innervating the head, the neck, the thoracic cage, the body wall being skin, musculature, bone, and the limbs, arms, legs, extremities. So head, neck, thoracic cage, body wall, limbs. Okay, sympathetic chain. All right, let's look at our next beautiful picture. All right, this one is collateral. Collateral means to the side. Now, this one is a little bit harder to illustrate. In the textbook, they take the image and they flop it to the side because realistically, our collateral prevertebral ganglia are located in front of the spinal cord. So you have celiac here at the top, and then you have the superior mesenteric, and then you have the inferior mesenteric moving down towards your pelvic region, okay? Celiac is top of the stomach, so that's the top of the abdominopelvic region. Superior mesenteric is, you know, moving down to basically like the small intestine and a little bit into the large intestine area. And then inferior mesenteric is the lower pelvic region. So essentially the collateral prevertebral ganglia are innervating, those postganglionic neurons are innervating your guts, your organs in the abdominopelvic cavity. Remember, our sympathetic chain did the body wall. So that's going to be the skin and the muscle and the bone surrounding the abdominopelvic cavity. And the collateral is going to do the inside, the organs of the abdominopelvic cavity, the guts. Okay. We still have a short preganglionic neuron fiber and a long postganglionic or ganglionic neuron fiber, okay? And then finally, we have, let me go ahead and, you go ahead and edit in your notes. This should be adrenal medullae with an AE ending because I've drawn in both. Okay, so the adrenal gland is this little triangular shaped gland sitting on top of your kidneys. You have two kidneys, you have two adrenal glands. Now, it's not obviously that huge in comparison to the kidney, and it's not quite that triangular in shape, but okay, my skills are limited. Alrighty. Now, the medulla is going to be talking about the inside of the adrenal gland. So I've drawn a smaller triangle inside. Now, in this case, we're going to see a few exceptions. So one major exception is that preganglionic neuron fiber is extending all the way from the spinal cord to the adrenal gland. So it's long, and we didn't see that before. It was short before, but now it's super duper long. And notice how teeny tiny my ganglionic neuron is that I've drawn in. Okay, so now it's flip-flopped. We have a long preganglionic fiber and a short ganglionic fiber. But here is the other really major thing. Here we go. Those ganglionic neurons are releasing the neurotransmitter into the bloodstream. And so now, instead of it being a chemical messenger released at a synapse, which is what a neurotransmitter is, the chemical is instead released directly into the bloodstream. So it is now acting like a hormone. Hormones are chemical messengers that are transported through the bloodstream and have more of a longer lasting widespread effect. Okay, now the neurotransmitters or chemicals that are released here are epinephrine and norepinephrine. So you more readily know epinephrine as adrenaline. So this is why you can have that experience of driving down the car. You look up and you see that the cars in front of you have stopped, so you slam on your brakes super fast. You're safe. The car in front of you is safe. The folks behind you are safe, so no damage is done. But for like 10 or 15 minutes, your heart is still racing and you're still in sympathetic activation. So that very quick rapid response is the nervous system control of your muscle, right? And then that kind of longer lasting response is because you have flooded your body with epinephrine and norepinephrine here at the adrenal medullae. So taking a look here, this is, oops, move this over back to this slide. Here is again that same summary that we saw at the beginning in our conversation about the organization. Short preganglionic fibers located between T1 and L2 of the spinal cord. 3 different types of ganglia, sympathetic chain, either side of the vertebral column, innervating the head, the neck, the thoracic cavity, the body wall, the limbs. And then the collateral, which is, remember, right in front of the spinal cord. So you have celiac, superior mesenteric, inferior mesenteric, innervating the organs inside that abdominopelvic cavity. And then our adrenal medullae, AE ending, would be the plural form, which is acting differently from the others. Long preganglionic fibers, short ganglionic fiber, release of that neurotransmitter into the bloodstream, so now it's acting like a hormone. And then my final little note here I've made is that long ganglionic fiber. Now notice here I've written preganglionic fibers above ganglia and then ganglionic fibers below. And the reason here is, of course, that transition of the signal goes through the ganglia to get from the preganglionic fiber to the ganglionic fiber. All right, next let's look at, what does it mean when we say the sympathetic is fight or flight? What is the sympathetic actually signaling for the body? What is its function? So it's going to increase our alertness, increase our heart rate, blood pressure, increase respiration rate and depth of that respiration. It's going to actually decrease your energy reserves. It's going to mobilize those energy reserves. So if we're mobilizing it, we're breaking down the triglycerides to release fatty acids into the bloodstream. So we're getting rid of those reserves. And then also we're going to break down the polysaccharide glycogen in our liver cells and go ahead and convert that into glucose. So we're having a lot of fatty acids and glucose available for ATP production in our cells because we are, as you can see, increasing the metabolism of those cells. And then we'll also see an increase in our muscle tone so that our body is really like tensed and ready to respond to, you know, whatever this stressful situation is. Okay, so here this makes sense why we would call it the fight or flight response. All right. Next, we need to look at what are described as our neurotransmitters. So we have already described neurotransmitters as those chemical messengers that are you know, released at a synapse. And so it's a very localized sort of, very rapid sort of communication. Now, when we're talking about the sympathetic division, the primary neurotransmitters involved are epinephrine, which will be abbreviated just capital E, and norepinephrine, which will be abbreviated big N, big E. So epinephrine and norepinephrine are the primary neurotransmitters released by the sympathetic division. We already sort of talked about them as being released from that adrenal medullae and then flooding the body as hormones moving through the bloodstream. In addition, we will see our old friend, big A, big C, little h. Do you remember what neurotransmitter that is? Hey, good recall, acetylcholine. Acetylcholine is going to be released by all of those preganglionic neurons. But then we will also see acetylcholine released by some of our ganglionic neurons. So you'll see acetylcholine released at every preganglionic, ganglionic synapse, and then also some of those ganglionic neurons will release acetylcholine. And then another neurotransmitter that we'll see released is nitric oxide, big N, big O, which we'll talk about a little bit, not a whole lot in chapter 16, but we will in later chapters. Now, as we are again talking about the primary neurotransmitters of the sympathetic division are, of course, norepinephrine and epinephrine. In order for those neurotransmitters or chemical messengers to, you know, activate that cell, it has to bind to an adrenergic receptor. So any synapse that involves the release of norepinephrine and epinephrine are called adrenergic synapses. And so any receptor that binds to norepinephrine or epinephrine are called adrenergic receptors. Now remember, I mentioned epinephrine is more maybe commonly referred to or more familiar to you as adrenaline. So that's how we're getting adrenergic. So think about that connection. Now, there are two different types of adrenergic receptors. There are alpha and beta receptors. So when we're talking about the sympathetic division, cells that respond to norepinephrine and epinephrine will have to have either alpha or beta receptors as part of their cell membrane. Now, when we are talking about the relationship of these neurotransmitters to these receptors, we see that norepinephrine most often binds to alpha receptors. Although in some cases norepinephrine will bind to beta, but think about it as more often binding with alpha. In the case of epinephrine, we will see it binding to both, so we will get more of a generalized activation of the sympathetic division with the release of our epinephrine. Now, the key thing to pay attention to as we go forward in our discussion of receptors is that there are different types of receptors, and the receptors themselves are what determines whether the cell is stimulated or inhibited by norepinephrine and epinephrine. So norepinephrine and epinephrine can bind to alpha and beta receptors, and then depending on which type of receptor it is, the cell will either be stimulated or it will be inhibited, okay? Now, as we talk about these chemicals, norepinephrine and epinephrine binding to our receptors, we'll see that our alpha and our beta receptors both are using what's described as a G protein system and also a second messenger. And so what we see is that your norepinephrine would bind to the receptor, and this would activate a G protein. Now, the G protein then triggers a second messenger. The second messenger is what triggers the change in the cell. So the norepinephrine and epinephrine don't actually move into the cell. Their receptors are not actually opening up and allowing an ion to move through like what we saw in AMP1. Instead, we see this G protein being activated, triggering a second messenger. Very often those second messengers are cyclic AMP, so that's abbreviated C, little c, big A, big M, BP, or calcium ions are often another example of a second messenger. Now for me personally, I like to think of my G protein as sort of like the butler answering the door. So your norepinephrine binds to alpha, that's sort of like it's knocking on the door. The butler opens the door, and norepinephrine is basically like, hey, I'm here. And so the butler turns and hollers at the second messenger, activating the second messenger, who then is going to make the actual change happen in the cell. So that's sort of how I think about my G protein and second messenger system. I don't know why I pick a butler. I mean, anybody can answer the door, right? Okay. Well, we're talking about our alpha and beta receptors. Let's look at alpha first. There are two different types of alpha receptors. So alpha 1 is often located on smooth muscle, and when your epinephrine binds to your alpha 1 receptor, this is going to excite your smooth muscle. So that's going to cause contraction of the smooth muscle. Alpha 2 receptors are located on both preganglionic sympathetic neurons and parasympathetic neurons, and it's inhibitory. So if you get a release of your epinephrine, not only are you going to maybe be stimulating smooth muscle, but by binding and inhibiting the parasympathetic neuron, you're going to be coordinating your two divisions. Because remember, they have opposite actions. So if you need to be able to stimulate a tissue with the sympathetic division, you need to then stop any signaling that might be coming, contradictory signal that might be coming from the parasympathetic. Okay, so here we're seeing like epinephrine is the same hormone that's released and binding. So epinephrine can act either excitatory or inhibitory depending on whether it bound to an alpha 1 or an alpha 2. So the type of receptor is what's really important here in determining how the cell responds to that signal. Beta receptors, there are 3 types. So beta 1 is going to stimulate skeletal and cardiac muscle. So that's going to be involved in that muscle tone and that increasing heart rate. Beta 2 is going to be involved in inhibition of smooth muscle, so that's going to allow it to relax. So if we want our blood vessels to dilate instead of being constricted, we need to open up that vessel, and so we need it to relax. That would be a beta 2. And then your beta 3 receptors are located on your adipocytes and are going to be involved in that releasing of those energy reserves, so that breaking down of those triglycerides to release fatty acids. So again, here we can see norepinephrine being released, and depending on whether the cell has a beta 1, a beta 2, or beta 3, we're going to get it to either stimulate or inhibit that cell. So again, one of our major takeaways here is that the neurotransmitters themselves can be either excitatory or inhibitory. It's dependent on the type of receptor that the cell has, and the cell gets to choose that, right? So the nature of the cell is determining what types of receptors it's making. And that's it.